The world of medicine has seen its share of miracle cures, from the polio vaccine to heart transplants, but all past achievements may pale in comparison to the work of Dr. Alice Crippen. Thank you so much for joining us this morning.
Not at all.
So, Dr. Crippen, give it to me in a nutshell.
Well, the premise is quite simple. Take something designed by nature and reprogram it to make it work for the body rather than against it.
You're talking about a virus?
Indeed, yes, in this case the measles virus, which has been engineered at a genetic level to be helpful rather than harmful. And I find the best way to describe it is if you can imagine your body as a highway and you picture the virus as a very fast car being driven by a very bad man, imagine the damage that car could cause. But then if you replace that man with a cop, the picture changes, and that's essentially what we've done.
Now, how many people have you treated so far?
Well, we've had 10,009 clinical trials in humans so far.
And how many are cancer-free?
10,009.
So you have actually cured cancer?
Yes, yes. Yes, we have.
Singapore has reached a new milestone in its biomedical efforts. An antibody-drug conjugate called EBC129 will be the first made-in-Singapore antibody-drug conjugate to be approved to enter clinical trials by the US Food and Drug Administration. Now, this drug is able to selectively target cancer cells across a range of solid tumours, killing it while sparing normal, healthy cells.
And here to tell us more is Dr. Andre Chu. He's Deputy Executive Director at A-STAR's Bioprocessing Technology Institute. And, of course, Dr. Veronica Diemeyer, Asset Development Leader at the Experimental Drug Development Center.
Hello.
Let's start with Dr. Chu first. And a very simple question. We just heard Dawn describe that. It targets specific cancer cells and does not indiscriminately kill healthy cells. How does that work?
Well, thanks, Dawn. Thanks, Wisu, for having us on the program. We are very excited for this opportunity to share about the development of EBC129. So, antibody-drug conjugates, or we call it ADC in short, is composed of an antibody and is linked to a drug compound. So, it's a new and exciting class of biopharmaceutical that is highly potent and is designed as a targeted therapy for treating cancers while sparing, as you said, normal and healthy cells. So this is unlike conventional treatments such as chemotherapy, which is non-specific. So how does it work? The antibody portion, like a magic bullet, a term that was coined over 100 years ago, will specifically home and bind onto markers that are present on cancer cells and delivers this toxic payload to kill it. So in short, if it's a cancer cell, the ADC will find it and kill it.
Right. So it's very targeted in its approach. Dr. Diemeyer, let's bring you in on the conversation. Tell us about that developmental process, what was involved in it. I think people will be curious about this whole ECB129 and what makes 129 specifically, part of the story. And tell us about that process.
Yeah. So it all started out as a collaboration. It's between the Bioprocessing Technology Institute of A-STAR and the National Cancer Center. They have discovered this antibody and then have brought the Experimental Drug Development Center, EDDC, in as a national platform for drug development to help develop this antibody. And in the process, we have engaged another A-STAR research institute, the Institute of Molecular Cell Biology, to help us with a particular assay that can select the patients to develop that part of this assay for the patient selection. So it's a very collaborative effort to develop this ADC, and the whole process takes a very long time. So it's about 18 months from the lead candidate until when we have the product that can go into man.
Right. Dr. Chu, you've got the first sort of trial, the first phase of the trial, and then you'll have, potentially identifying patients and so on. The interest will be how soon, perhaps? I mean, what would the timeline be if all goes well and you get the correct sort of success markers that you need, how soon might this be something that would be practically used?
Veronica, I think you might be better at this question, actually. Yeah.
We should be able to say probably after 5-6 months whether we have a drug that can help to shrink tumors in patients. Because we are lucky in this case because this ADC has a toxin that is very known how it works. And the dose that is required from this toxin is also known. And so if we can reach this dose level, we are likely going to have success in the clinic. And so we know how many levels we have to escalate by until we reach this dose level.
Oh, thanks so much for coming in, both of you. Dr. Andre Chu from A-STAR's Bioprocessing Technology Institute and Dr. Veronica Diermeier from the Experimental Drug Development Center. Thanks so much for coming in this evening.
Thank you.